An expert panel reviewing the published evidence on protein requirements in older adults concluded that the current official recommended dietary allowance of 0.8 grams of protein per kilogram of body weight per day is not sufficient for adults over 65. The PROT-AGE study group, convened specifically to address this gap, recommended 1.0 to 1.2 grams per kilogram for sedentary older adults, and 1.2 to 1.5 grams for those who are physically active. For a 75 kilogram adult, that difference between the official RDA and the PROT-AGE recommendation could be 30 grams of protein per day, roughly the amount in a large chicken breast. Most adults over 55 are unknowingly operating below what the evidence suggests their muscles actually need. Why does this matter so concretely? Skeletal muscle is not a cosmetic tissue. It is metabolically central to how the aging body functions. Muscle mass determines your resting metabolic rate. It is the primary site where glucose is cleared from the bloodstream after meals, making it central to insulin sensitivity and metabolic health. It protects bones by providing the mechanical loading that prevents osteopenia. It is the buffer that keeps a trip on the sidewalk from becoming a fall with a fractured hip. In the 5 years following a hip fracture in adults over 65, mortality rates are estimated at 20 to 30%. Muscle is not something to optimize for aesthetics at 55, it is something to preserve as a survival asset. The official RDA of 0.8 grams per kilogram was established not as an optimal target but as the minimum requirement to prevent nitrogen deficiency in a general adult population. That population was largely studied in younger adults. Applying a minimum to prevent deficiency standard to a 60-year-old who was actively trying to preserve muscle mass through the decade of peak sarcopenia risk is a category error and one that has real biological consequences for anyone following it uncritically. In this video, we will examine why the official protein RDA falls short for this age group, what the PROT-AGE evidence review concluded about optimal intake, why the timing and distribution of protein across the day matters as much as the total amount consumed, which protein sources are most effective at triggering muscle protein synthesis in older adults, and how kidney function changes the calculation for a subset of this population. Quick note before we begin, this is educational content based on published research, not medical advice. Talk to your physician before changing your regimen. Now, the study. Muscle mass peaks in most adults somewhere between the ages of 25 and 35, and begins a slow but measurable decline thereafter. After 50, that decline accelerates. The clinical term is sarcopenia, the age-related loss of skeletal muscle mass and strength, and it is listed by the World Health Organization as a recognized disease entity. By the time a person reaches 70, they may have lost 20 to 30% of the muscle they had at their peak. By 80, in sedentary adults, the losses can exceed 40%. This is not a cosmetic concern or an athletic one. Muscle mass is functionally linked to resting metabolic rate, insulin sensitivity, bone density, fall prevention, immune regulation, and all-cause mortality. The studies examining the relationship between muscle mass, grip strength, and longevity are as consistent as any body of evidence in gerontology. What makes the protein story particularly important for adults over 55 is a biological phenomenon called anabolic resistance. In younger adults, a modest dose of dietary protein, say 20 to 25 grams of high-quality protein, is sufficient to maximally stimulate muscle protein synthesis, the cellular process by which muscle tissue is built and repaired. In older adults, the skeletal muscle responds less efficiently to the same protein stimulus. The molecular mechanisms include blunted mTOR signaling, reduced sensitivity of the aminoacyl tRNA synthetase pathway, and splanchnic sequestration, the increased extraction of dietary amino acids by the gut and liver before they reach the peripheral circulation. The practical result is that older adults need a higher dose of protein, delivered more strategically, to achieve the same muscle protein synthesis response that a 25-year-old gets from a more modest intake. The original recommended dietary allowance for protein was established in nitrogen balance studies conducted primarily on younger adults. It was set at 0.8 grams per kilogram per day as a minimum to prevent nitrogen deficiency in a general population, not as an optimal target for preserving skeletal muscle mass in a 65-year-old experiencing anabolic resistance. This distinction between preventing deficiency and optimizing function is one of the most consequential gaps between official nutritional guidelines and what the current mechanistic and clinical evidence actually supports for older adults. The PROT-AGE study group was convened specifically to re-examine that gap, bringing together an international panel of geriatric nutrition and muscle physiology experts to synthesize the best available evidence and produce updated recommendations for older populations specifically. A position paper published in the Journal of the American Medical Directors Association by Bauer and colleagues from the PROT-AGE study group synthesized the available evidence on protein requirements in older adults and produced evidence-based consensus recommendations. The PROT-AGE group was an international panel of experts in geriatric nutrition, muscle physiology, and clinical dietetics, including researchers from Europe, North America, and Australia, convened specifically to produce the most rigorous and comprehensive review of protein needs in aging to that point. The review covered multiple independent streams of evidence rather than relying on any single study type. Nitrogen balance studies provide information about minimum protein requirements to prevent deficiency. Acute protein kinetics research, using stable isotope tracer methodology, which allows researchers to measure the rate of muscle protein synthesis in real time following a meal, provides mechanistic insight into how different doses and sources of protein affect the synthetic response. Epidemiological cohort studies show what protein intake patterns are associated with better preservation of muscle mass and physical function over years. And randomized intervention trials test whether higher protein intake actually produces better functional outcomes when implemented prospectively. The panel distinguished carefully between subgroups within the older adult population, recognizing that protein needs are not monolithic across 65-plus adults. A healthy, sedentary 68-year-old has different requirements than a 78-year-old recovering from a hip fracture or a 62-year-old doing resistance training 3 times per week. The PROT-AGE recommendations were structured to acknowledge this heterogeneity. What the researchers found when they examined the acute protein kinetics literature was particularly instructive and mechanistically central. Studies using isotope tracer methodology consistently showed that older muscles require higher per meal protein doses to achieve the same anabolic response as younger muscles, the direct experimental demonstration of anabolic resistance. This biological reality, reproducible across multiple research groups using different isotope tracers and different protein sources, was the foundational finding that made the inadequacy of the standard RDA scientifically clear. The panel also reviewed evidence on protein quality, specifically the role of essential amino acids and the branched-chain amino acid leucine in initiating muscle protein synthesis at the cellular level. The evidence consistently pointed toward leucine as the primary trigger for the mTOR-mediated anabolic signaling cascade, and toward protein sources with higher leucine content showing greater anabolic efficiency per gram of protein consumed. The PROT-AGE panel's primary recommendation was that older adults, defined as those 65 and above, require 1.0 to 1.2 grams of protein per kilogram of body weight per day to maintain muscle mass and physical function during normal health. For physically active older adults, those engaging in regular aerobic or resistance training, or those experiencing acute or chronic illness, recovery from surgery, or hospitalization, the recommendation rose to 1.2 to 1.5 grams per kilogram per day. These are not arbitrary numbers. They are the consensus thresholds derived from the isotope tracer literature, adjusted for the degree of anabolic resistance documented in older muscle tissue. To put those numbers in concrete terms, a 70-kilogram adult following the standard RDA of 0.8 grams per kilogram would consume 56 grams of protein daily. The PROT-AGE recommendation for the same person at 1.2 grams per kilogram is 84 grams, a 30-gram difference, every single day. Over the course of a year, that cumulative difference in the protein signal delivered to aging muscle is not negligible. It represents a sustained biological environment that either supports or undermines the cellular machinery responsible for maintaining lean tissue. But that's not where the story ends. The panel found that total daily protein intake, while necessary, is not sufficient on its own. The distribution of that protein across the day is equally important, and this was one of the most practically significant findings in the review. The evidence from acute protein kinetics research pointed toward a threshold of approximately 25 to 30 grams of high-quality protein per meal as necessary to maximally stimulate muscle protein synthesis in older adults, accounting for anabolic resistance. Below this threshold, the synthetic response is submaximal. The meal does not fully trigger the anabolic signal. Most adults in Western dietary patterns consume very little protein at breakfast, a moderate amount at lunch, and the large majority of their daily protein at dinner. This skewed distribution, even when total daily intake is adequate, may result in only one meal per day, dinner, delivering the threshold dose needed for maximal synthesis. The panel concluded this distribution pattern is suboptimal for preserving muscle mass, and that spreading protein more evenly across 3 meals, each targeting the 25 to 30 gram range, is a meaningful practical improvement. What the researchers found next is even more important for this age group. Pre-sleep protein consumption, specifically a dose of approximately 40 grams of a slowly digested protein such as casein, was supported by emerging evidence at the time of the PROT-AGE review as a strategy for maximizing overnight muscle protein synthesis. During sleep, the body enters its longest daily fasting period, and providing a slow-release protein source before sleep may extend the anabolic window into the overnight hours, supporting net muscle protein balance during a period that is otherwise catabolic. The limitations of this evidence deserve honest examination. The PROT-AGE recommendations are based on a synthesis of studies that varied considerably in methodology, population, and duration. Many of the acute protein kinetics studies that provide the mechanistic foundation, the isotope tracer experiments showing anabolic resistance, were conducted over hours to days in controlled laboratory settings, not months in free-living adults. The long-term question of whether consistently maintaining protein intake at 1.2 or more grams per kilogram translates to measurably preserved muscle mass, functional outcomes, and mortality reductions across years of follow-up is supported by epidemiological evidence and a growing body of randomized trial data, but has not been definitively settled in single large-scale, long-duration randomized controlled trials powered for functional endpoints. There is also ongoing scientific debate about the upper bound of per-meal protein utility for synthesis. Some researchers, drawing on dose-response kinetics data, argue that the muscle protein synthesis response plateaus at approximately 40 grams per meal for older adults, and that exceeding 1.6 grams per kilogram of total daily intake produces diminishing returns for muscle outcomes specifically. Others argue that higher intakes may be beneficial, particularly during caloric restriction, illness recovery, or extreme physical stress. The PROT-AGE range, 1.2 to 1.5 grams per kilogram, sits within the well-supported range and below the area where the evidence becomes genuinely contested. The limitations of this evidence deserve particular attention regarding kidney function. A long-standing clinical concern has been that elevated protein intake accelerates the progression of kidney disease by increasing glomerular filtration pressure. The current evidence in adults with normal kidney function, defined as eGFR above 60 milliliters per minute per 1.73 square meters, does not support the conclusion that protein intake in the 1.2 to 1.5 gram per kilogram range causes harm. But in adults with existing chronic kidney disease, the picture is meaningfully different. For those individuals, higher protein intake does require careful management, individualized targets, and physician and dietitian oversight. This is not a recommendation that applies uniformly across all older adults. It is precisely the reason to include kidney function markers, specifically eGFR and urine albumin-creatinine ratio, in any conversation about increasing protein intake with a physician. One further note of reassurance about the recommendations themselves: the PROT-AGE conclusions have been broadly affirmed by subsequent expert bodies, including the European Society for Clinical Nutrition and Metabolism and the International Osteoporosis Foundation, which have published overlapping consensus recommendations for older adults. The convergence of independent expert groups on a similar target range lends these recommendations greater credibility than any single position paper alone. 3 takeaways grounded in what the PROT-AGE evidence actually tells adults over 55. Takeaway 1: establish your actual current protein intake before deciding how much to increase it. Most adults over 55 significantly overestimate their protein consumption when they have not tracked it systematically. 3 to 5 days of honest food tracking using any standard nutrition app typically reveals a gap between perceived and actual intake. Once the baseline is known, work with your physician to determine whether increasing toward 1.2 grams per kilogram of body weight is appropriate, particularly given your kidney function status. For a 75 kilogram person without kidney disease, that target is 90 grams per day, attainable from whole foods, but requiring deliberate attention to protein at every meal, not just dinner. Takeaway 2: restructure protein distribution across 3 meals, targeting at least 25 to 30 grams per meal. A typical Western breakfast of coffee and toast delivers 5 grams.
A lunch salad with chicken provides 15. A larger dinner provides 50.
Even if daily total reaches 80 grams, only dinner delivers the threshold dose for maximal muscle protein synthesis. The fix, protein anchor breakfast with eggs, yogurt, cottage cheese, or a high-quality shake, and ensure lunch reaches 25 grams. Both meals then contribute to the cumulative synthesis signal. Takeaway 3, prioritize leucine-rich protein sources, particularly around exercise. Leucine is the primary molecular trigger for the mTOR signaling pathway that initiates muscle protein synthesis. Animal proteins, whey, eggs, beef, chicken, fish, are generally rich in leucine relative to most plant proteins. Whey in particular has among the highest leucine concentrations and is rapidly digested, well suited to post-exercise consumption. There's a catch that matters specifically if you're over 55, if you have kidney disease, elevated creatinine, or an eGFR below 60. Discuss protein targets with your physician and a registered dietitian before making significant changes. The evidence reviewed by the PROT-AGE study group makes a compelling case that 1.2 grams of protein per kilogram per day is a more appropriate target for adults over 65 than the official RDA, with higher amounts supported for those who are physically active. The distribution across 3 threshold meals matters as much as the daily total. And leucine-rich protein sources deliver a more potent muscle protein synthesis signal per gram consumed, a distinction that matters more after 55 than at any earlier age. Preserving skeletal muscle through the 6th and 7th decade is not an athletic goal. It is one of the most evidence-supported longevity interventions available. If these numbers feel abstract, the PDF guide on reading your biomarker panel, linked in the description, includes the key markers for tracking muscle and metabolic health alongside your standard blood work, including the kidney function markers relevant to determining your appropriate protein target. Subscribe to stay current with the evidence as it continues to develop in this fast-moving field. This is educational content, not medical advice. Always consult with your physician before changing your regimen. Studies are preliminary, individual responses vary.